Up muscles. We're coming toward the end of the course. There's much fewer assignments. Just make sure you turn in uh, any question. Guided notes. Let's look at the PowerPoint for muscles and let's go through that again. There's some difficult concepts in here. So we're presenting now. Good. Okay, so muscles are either voluntary, like skeletal muscle, and muscles otherwise are involuntary, like cardiac and smooth muscle. You don't have to do anything. Your heart beats automatically. I'm happy mine is continuing to beat. I'm 74 years old, but I'm still kicking, and I have a new granddaughter. So, again, all your skeletal muscles, like your biceps, triceps, uh, trapezius, uh, adductor magnus in the leg and the thigh, gastrocnemius in the calf, those muscles are all voluntary and you have to control them. Skeletal muscles are attached to bones. They produce movement, maintain posture and body position, support soft tissues like the lungs and the heart, uh, guard entrances and exits, and maintain body temperature. So they guard your oral cavity and they guard the part of the end of your digestive system where you get rid of waste. So let's look at the structure of muscles. All muscles will have uh, an epimysium, which is the connective tissue around the entire muscle. Each fascicle, each fascicle will be surrounded by a perimysium, and each muscle fiber will be surrounded by an endomysium. So connective tissue holds the muscle in place, surrounds the entire muscle. So one muscle is an organ. A muscle is a bundle of fascicles, bundles of muscle cells. The endomysium surrounds each muscle fiber, a cell. The epimysium surrounds the whole muscle, and perimysium is around each fascicle. So let's move on to a picture. So here's an image. So the entire muscle. This right here is a single muscle fiber or a cell. So it's surrounded by a sarcolemma and uh, will be surrounded by endomysium. So T tubules allow electrical signals to get into the muscle. Around some T tubules are uh, a triad, which has two uh, terminal cisterna and one T tubule. So the sarcoplasmic reticulum is this network of branched or storage sacs that hold calcium. If you look at an individual muscle fiber called a myofibril, it's going to be made up of an I band and an A band. The thick filaments are called myosin, the thin filaments are called actin. You can see that the actins are attached to the Z line. And if you look at a cross section, you can see that all of the thick filaments, the myosin filaments, are surrounded by thin filaments. So something's going to happen to allow each sarcomere from Z line to Z line to shorten. So each muscle fiber has a Z line where the actin is attached, an M line in the middle of the sarcomere. Uh, the myosin fibers are held, or filaments are held in place partially by the M line. Each thick filament will have a myosin head and a myosin tail, and these are able to attach to the thin filaments at the active sites. We'll get into how the contraction works in a moment. So we look at a relaxed muscle, Z line to Z line, you can see that the actin is attached to the Z lines, but the actin only overlaps with the myosin thick filaments only in a partial overlap. If once the muscle contracts, you can see that the actin has been pulled into the center of the sarcomere, and the whole sarcomere has contracted. And once the I bands are much smaller, the A band is the same distance, but the contraction of the sarcomere happens when the myosin pulls the actin into the center of the sarcomere. Each muscle has a nerve. So each muscle filament or muscle fiber has a neuromuscular junction. That's the junction between the axon and the axon endings. So that's called the neuromuscular junction, where nerve endings attach to the muscle just above well, they don't attach to the muscle. They're just above what's called the motor end plate. So if you look at the axons, you have the synaptic terminals, because this is a synapse between muscle and nerve. A synapse is the gap between the nerve and the muscle. Within the synaptic terminals, there's vesicles containing acetylcholine. When the action potential or nerve impulse comes from the spinal cord and terminates in the synaptic terminal, these vesicles are going to merge with the membranes of the axon terminals or synaptic terminals, release acetylcholine. Acetylcholine is going to bind to the sodium chemically gated receptors, which opens up sodium channels. And then once those sodium channels enter, will follow, contraction of the muscle will follow. Immediately, acetylcholinesterase will break up the acetylcholine so the muscle can relax. So the contraction process, I said that the active sites on actin have the cross bridges attached to the actin active sites. The thin filaments are pulled into the center of the sarcomere by the attachment and pivot, and then detachment and return of the myosin heads. So how's that work? So the resting sarcomere has tropomyosin covering the active sites. The troponin is the location where calcium is going to bind to the troponin and cause a rotation of the tropomyosin. The energized myosin heads, ATP, has bound to the myosin heads and dissociated into adenosine diphosphate and phosphate, and with the calcium allowing the myosin heads to attach to the binding sites on the actin, the myosin heads move forward, ratchet or pivot toward the center of the sarcomere that pulls in the actin fibers. As soon as that happens, the myosin heads detach from the actin myosin sites. ATP then recharges the energy and cocks the myosin head. And as long as there's ATP and calcium is present, the contraction will follow the pivoting of the attached myosin heads. And then as long as there's ATP around, the muscle can contract. So we have somebody coming in here. Okay. There we go. Everybody's in. Okay, so back to the PowerPoint. Okay, so we keep going. Here's a summary again of the contraction process. So every skeletal muscle has to have an axon terminating at the synapse. The synapse is the gap between the uh, synaptic terminal and the motor end plate. So acetylcholine is released, the sodium rushes in, the action potential causes sodium to continue to move into the T tubules, and it causes the sarcoplasmic reticulum to release calcium. Calcium binds to the troponin. It causes the tropomyosin to rotate, and the actin binding sites have the myosin heads attach, and the myosin on either side of the the actin on either side of the myosin has the uh, myosin heads pivot and pull the uh, actin to the center of the sarcomere. Then acetylcholine is going to break up acetylcholine. So acetylcholinesterase is uh, an enzyme that breaks down acetylcholine. Once the channels are closed again, the active sites are covered on the actin and the muscle relaxes and contraction ends. Okay, let's keep going. Let's shut down the recording for a moment and then keep recording. Stop recording, stop.